Hi everyone, welcome to my channel The Med for All. Uh, Today we are going to start digestion and absorption of carbohydrate. So previous video, my first video of biochemistry, we discussed about the definition of carbohydrate, classification of carbohydrate, function of carbohydrate. Okay, so today I'm making one separate video that is about digestion and absorption. So let's start. If you see here, the meaning of digestion, the word only indicate that breakdown. Okay, so the all larger food, the larger food means our body cannot utilize the larger or complex food. It should be converted into smaller form. Okay, all those foods should be converted into very smaller form or simpler form, so our cell tissue can utilize this. So that is called the digestion. So mainly the carbohydrate which we are taking from diet, mainly that is a polysaccharide. So most of the food mainly like rice we are getting carbohydrate, which mainly found, that is a starch, that is a polysaccharide. So you can see polysaccharide is a larger molecule, larger carbohydrate. Another you can say cellulose, mainly the dietary fiber which our body cannot able to digest. So mainly starch, glycogens are mainly the stored form of carbohydrate from animal. Okay, found in the animal's tissue. So polysaccharide, glycogens, okay, these are the polysaccharide and larger molecule. It should be converted into the smaller form. So always remember digestion takes place in our GI tract, mainly in the gastrointestinal tract, either in the stomach or sorry, either in the mouth, stomach or small intestine. But carbohydrate digestion starts from small intestine, that also we are going to discuss. So carbohydrate which are found in our diet, that is called, first one is called polysaccharide. Either it may be the starch or glycogen. Disaccharide, as you know that many already we discussed in classification, having two monosaccharides, that is called disaccharide. So lactose, which is called the milk sugar, maltose, sucrose. So these are the disaccharides. Monosaccharide no need to digest the monosaccharide because monosaccharide is a smaller molecule. So further hydrolysis or breakdown of monosaccharide is not possible at all. For this reason, our body can utilize glucose, fructose, and galactose. These are the monosaccharides. So this is about the okay. So all these carbohydrate digestion take place in GI tract. Always remember. Now we'll see that details about the carbohydrate metabolism. So how metabolism starts. So two types of enzymes are there. There are two enzymes are there. If you see here in this video, it indicates that there are two enzymes are there. Okay. So enzymes which are helping for digestion. Just to see this point, the enzymes mainly important enzymes of carbohydrate. One is called amylase. The amylase enzymes are mainly found in our saliva also. It is found in our pancreas, mainly pancreatic juice also. So these enzymes, okay, helping the breakdown of polysaccharide into disaccharide, or you can say into the oligosaccharide. So that is called the the polysaccharide is mainly which consists more than we discussed, it consists more than 9, 10 monosaccharide combined together. So with the help of amylase, the amylase enzyme, okay, this is the enzyme found in saliva, okay, found in pancreatic juice. So mainly always remember carbohydrate digestion starts from the mouth due to the presence of salivary amylase, whereas completed mainly in small intestine because in our mouth the food and saliva gets very lesser time after that. After the chewing of food, when you swallow the food, food enters into the stomach. So in stomach mainly we know that the pH of stomach is acidic in the nature. Around you can say that very highly acidic in the nature. So in acidic pH, amylase activities are lost. So okay, so amylase is only active in alkaline pH. Always remember amylase enzyme, okay, which is active only in alkaline pH. So our stomach is acidic pH, so the activity is lost. So you can say that digestion of carbohydrate in our stomachs are stopped. Then again pass into duodenum. Duodenum receive pancreatic juice, and again digestion starts. So once the so mainly always remember. So polysaccharides are converted into disaccharides, where it is mainly completed in the duodenum. So still carbohydrate digestion is not completed in even in duodenum also. Pancreatic amylase only breaking polysaccharide into disaccharide or oligosaccharide. So even disaccharides are also the larger molecule. So all these, so the disaccharide, monosaccharide, which are mainly finally absorbed, so mainly these disaccharide should be broken into maltose. So what is called there is called maltase enzyme. So maltase enzymes are mainly found in the intestinal walls, in our small intestine, mainly jejunum, ileum. The brush border of our small intestine, a consistent number of enzymes. That enzymes is mainly helping the final digestion of carbohydrate and like sucrase enzymes. Okay, sucrase is enzymes which are mainly helping for uh, digestion of a disaccharide called sucrose. Lactase is an enzyme which is helping for digestion of lactose. Lactose is a milk sugar. So this is the enzyme. So first enzyme, so mainly you can say that polysaccharide broken into disaccharide or oligosaccharide in duodenum. Then food pass into the jejunum and ileum, where the brush border or you can say the intestinal wall of jejunum and ileum consists maltase, sucrase, or you can say trihalases, okay, maltase enzymes, lactase enzymes, so which mainly helps the final digestion of carbohydrate. Moving to the next slide, just to see in the figure only. So we have a salivary gland. So as you know that salivary gland can secrete saliva. Okay, saliva consists salivary amylase. Okay, so this amylase is helping for digestion of. Okay, so mainly polysaccharides. So these polysaccharides are not digested completely. Only 5% digestion take place. Some carbohydrates are converted into disaccharide. Now food is passing. Just to see the food is passing mainly from food pipe and it goes into the stomach. So in stomach we discuss that pH is very highly acidic in the nature. pH is acidic in the nature. So due to the acidic pH, you can say that digestion becomes stopped here. Then again it passes into the after 3 to 4 hours when gastric juice, mainly the stomach consists gastric juice like pepsin, hydrochloric acid, mucus acid, foods are mixed here and pass into the duodenum. So our duodenum receives pancreatic juice. Pancreatic juice consists pancreatic amylase. Now again amylase convert all those polysaccharide into disaccharide. Always remember, it is mainly converted into disaccharide. Disaccharides are still larger molecules. So it should be finally digested. That digestion takes place in our ileum. Okay, that is mainly take place in our jejunum ileum. So you can see digestion in the mouth, first happening in the digestion in the mouth, then digestion in the stomach. Mainly hydrochloric acid is not helping digestion, but it hydrolyzes the carbohydrate, and digestion in the small intestine is mainly completed. Okay, moving to the next slide, if you see that uh, digestion of carbohydrate, okay, that start in mouth upon the contact with saliva during mastication. So what is the meaning of mastication? Mastication means the process of chewing, chewing up food with the help of teeth, mixing with saliva. Okay, so that is called mastication. So saliva contains okay carbohydrate break splitting enzymes called salivary amylase, also known as ptyalin. This enzyme is also called the ptyalin. So which mainly break the cooked starch into okay disaccharides. So this is very simple. So location first start from okay found in the mouth. Ptyalin is found in the mouth. So pH in our saliva is around 6.7, slightly acidic. Okay, so enzymes hydrolyze and mainly start the breakdown of a carbohydrate. So you can see that like starch, glycogen, dextrin, these are the larger polysaccharides which consist alpha amylase. So glucose, maltose, and maltodextrin, these are the smaller molecules. So all these larger molecules should be converted into smaller molecules, that is the digestion. Moving to the next slide, if you see that why digestion does not completed in our mouth, because shorter duration of food. So mainly food, okay, shorter duration of food in mouth. In our mouth, food remains around 2 seconds. So that time is not sufficient for digestion. So that digestion is incomplete. Okay, so then it passes into the stomach. So once it passes into stomach, there is no any enzymes to break all those glycosidic bonds. So what is glycosidic bond? The bond which is found between the monosaccharides. So there is no bond, so there no breakdown. But however, okay, hydrochloric acid is found in the stomach, which is mainly helping the hydrolysis of mainly sucrose, fructose, and glucose. So sucrose is mainly disaccharide with the help of hydrolysis. Fructose and glucose are, but still this digestion is not completed. In the duodenum, we'll see that. So in duodenum what is happening? So this is called the duodenum. Just to see the figure here, this is known as the C-section duodenum, where pancreas is also found. Pancreatic juice are produced, exocrine part, and pass into the duodenum. So here mainly digestion takes place. Just to see, I am highlighting here in the duodenum. So mainly this part of our duodenum receives okay pancreatic juice, and digestion is mainly started here and completed also here. Mainly disaccharides are converted. So mainly the food bolus which reaches to the duodenum. So very simple. So the food bolus, just to see the food bolus reaches to the duodenum from the stomach. Okay, so mainly from the stomach, where it meets to the pancreatic juice. So mainly pancreatic juice consists carbohydrate splitting enzymes, and already we discussed that. That is called pancreatic amylase. So that is mainly the action is very similar to the salivary amylase, but it gets more time. So in stomach, no digestion, always remember, because of hydrochloric acid and pepsin. Again in duodenum start. Now moving to the next okay slide. So here alpha amylase enzymes are found. The pH of our pancreatic juice. Pancreatic juice is alkaline in the nature because of sodium bicarbonates are found. NaSCO3. Sodium bicarbonate is found in the pancreatic juice, so which maintain the alkaline pH. So in the duodenum, all those content which are coming from the stomach become neutralized and amylase action start the breakdown, so fully converted into disaccharide. You can see here it is fully converted into disaccharide. So all those amylase is mainly converting polysaccharide into disaccharide. Now you can see here the starch glycogen with the help of pancreatic amylase, which is converted into maltose, dextrin, oligodisaccharide. So mainly these are the disaccharide mainly. So where take place, this is a just you see leaf-like structure called pancreas. This is a duodenum. So take place here. Now moving to the next slide. So the main digestion take place again in the small intestine with the help of pancreatic amylase and completed by you can say that in our intestine mainly food stay for long period of time because of the length of our small intestine. If you remember that the length of our small intestine is around you can say that uh, around 7 meter long, so more time it gets. Okay, surface area is also high, so enzymes can act and help in the digestion. So already we discussed about the disaccharide. Okay, so all those disaccharides are mainly converted into our duodenum. Now we can see the enzymes. These are the enzymes. So these enzymes like maltose, okay, maltose, sucrose, these are the disaccharide. Lactose, these are the disaccharides. Maltose is mainly consisting glucose and two glucose molecules are connected together by glycosidic bond. So these maltose are mainly digested with the help of maltase enzyme. These maltase enzymes, where it is found? It is mainly found in our the brush border of small intestine. Sucrase enzymes are also found in the brush border of small intestine, mainly jejunum and ileum. Whereas the lactase enzymes is mainly found in the intestinal wall. So when lactase enzymes are absent, then patient will suffer from lactose intolerance test. So lactose is a milk sugar which is mainly found in mammalian sugar, a mammalian milk. So once if any patient having a deficiency, or you can say or lactase enzymes are absent, they cannot able to digest the milk. So lactase enzymes found in our intestinal wall that mainly break the lactose into glucose and galactose. So this is called the final pathway for digestion. So that is mainly completed in our you can say that jejunum and ileum. So by the end part of ileum, so mainly digestion is fully completed. Now absorption starts. So we discuss what is absorption. So very simple. Absorption means okay, the movement of the molecules or nutrients from digestive system. The movement, very simple. The movement of nutrients from digestive system to blood circulation. Mainly it pass into the blood circulation. So that is called the digestion. So very simple. So the movement of here you can see the movement of the nutrients from digestive system to blood circulation. There are three mechanisms are there which is helping for digestion. One is called passive diffusion. I hope everyone is knowing that passive diffusion. The meaning of passive diffusion is that the movement of molecules from higher concentration. Just see, this is called the intestinal wall lumen, and this is called the blood circulation, which is mainly found in submucosal layer. Their concentration of glucose or monosaccharide is higher. Now it is mainly moving from our intestinal lumen to the blood circulation. So this is called passive diffusion, which does not require any energy or carrier transport. Next one is called facilitated diffusion. Just you see here. Here we can see this is called facilitated diffusion. Facilitated diffusion means where some carrier molecules are required. These are the carrier molecules or protein which is helping to transport carbohydrate from higher concentration to lower concentration. So what is the difference between diffusion, simple diffusion, or you can say facilitated diffusion? In simple diffusion, no energy required, molecules are moving from higher to lower. But in facilitated diffusion, energy is not required, but it requires carrier protein, which are mainly found in our intestinal wall and help transport. The next one is called the active transport. Active transport means the movement of nutrients against the concentration gradient. Here totally opposite. The molecules are mainly moving from lower concentration to higher concentration with the help of sodium ATPase pump. So there is a sodium ATPase pump found on our you can say small intestine, the wall of our small intestine, and which mainly helping for you can say that helping for the digestion. Helping for absorption. So this is called the absorptions of carbohydrate. So this is called, you can see here the definition: down the concentration gradient, high or low, down the concentration gradient, high to low. But you can see the active transport means the against the concentration gradient from low to high. So no energy required here. Also energy not required, but here in active transport energy is required. Okay, so you can see carrier protein. If you see that passive does not require carrier protein, but if you see facilitated require carrier protein, active also require carrier protein. Here the slow action is very slow, movement is very slow in the passive, but in facilitated very fast. Even in active transport more faster. So in this way mainly the mainly glucose, or you can say the monosaccharide like glucose, fructose, galactose are mainly moved from small intestine to large intestine. That is called absorption. So I hope everyone is clear about absorption. So absorption takes place by three processes. One is called simple diffusion, where molecules like monosaccharides are moving from our intestinal lumen to blood circulation from higher concentration to lower without carrier molecule, without ATP. In facilitated diffusion, we discussed that where the molecules are moving from higher to lower, okay, without ATP but require carrier molecule known as protein. So some transporters are required which are helping to transport from higher to okay lower. Whereas active transport is totally opposite, where the molecules or monosaccharides are moving from lower concentration to a higher concentration, but it requires lots of ATP, sodium ATP from sodium exchange is there. So this is helping from absorption. So you have completed digestion and absorption of carbohydrate. These are the okay glucose transporter which is mainly found in our mainly SGLT, GLUT. These are the glucose transporter found in our intestine. Okay, so this is called absorption. So by this mechanism, so already we discussed that. So these are the transported found in our intestinal wall. Okay, these transporters are helping for absorption of monosaccharides. So we have completed. Okay, so this is a diagram how monosaccharides are absorbed. So you can see this is called that after digestion. Okay, so mainly with the help of simple epithelial tissue of our, you can say this is called the intestine. So cells mainly fructose or absorption mainly take place. Okay, in this way mainly take place from simple diffusion. Okay, so simple diffusion directly moving to the blood circulation. Facilitated diffusion, some carrier proteins are there. You can see here in the diagram very clearly mentioned. Okay, active transporter sodium ATPase pumps are there. So this is called the absorption of carbohydrate. So we have completed okay digestion and absorption. So next video I'll make one more video about the metabolism and disorder of carbohydrate metabolism, mainly diabetes mellitus, glucose tolerance test estimation. That is very important for okay from biochemistry. So, okay, so today only for this much. So thank you to everyone for watching my video. Please subscribe and like my video and give your valuable comment. Thank you. Have a good day.